Hello class, I'm here with the second half of the respiration review. In this case we're looking at the specific processes of respiration, including glycolysis and the Krebs cycle and oxidative phosphorylation, also known as the electron transport system. I will try to go through this as quick as possible, but take a look at the Prezi, which is linked back on the Ogden Wiki site, to take a look at some of the both some of the text and some of the diagrams, which can be helpful in being able to summarize the process for you. So first interesting thing is just to be able to understand the relationship between respiration and photosynthesis, in that they have this reciprocal relationship, in that the products of photosynthesis, which are glucose and oxygen, happen to be the reactants for the process of respiration. And so you have this cycle feeding in of one process feeding into another, and so this becomes a very helpful relationship in terms of maintaining balance between different types of both energy and matter on Earth. So to skip ahead a little bit, you can take a look at the Prezi. One of the things that we need is control, and control is necessary because the process of breaking down sugar can be somewhat dangerous. So you can take a look at that. But to move on with our study here, glycolysis begins the cycle. Glycolysis here on the left. The products of glycolysis are pyruvate, which go into pyruvate oxidation, which lead into the citric acid cycle. Electrons are going to be released through that process, going into oxidative phosphorylation, which will ultimately result in ATP. This is the biggest source of ATP here at the end, and that's really the end product for all eukaryotic cells. So we start with glycolysis. In this we start generally with glucose. It's going to be split in half. Pyruvate is what is created. You should know pyruvate, both its name and spelling. And it's going to create a little bit of ATP. It uses up 2, but produces 4, so we have a net of 2 ATP produced. And we also make a little bit, we also release some electrons, which are stored in NADH. It's a complex process. Here's a whole bunch of steps. Each of these steps has a particular enzyme that's responsible for helping to catalyze that reaction. 2 phases. The first phase is where that ATP is used up. The second phase is where 4 ATP are going to be formed along with those NADH, which are released from the electrons that are liberated in this process. So the net result is always something that's going to be particularly important. Before we get to that, glycolysis is universal. Every cell in every type of organism, from the simplest eukaryotic and eubacteria up to the most complex animals, all utilize glycolysis, and so this is something that really ties together all forms of life. So here's your inputs and outputs, so definitely take note of that. Now the products here have really one of 2 choices. In some organisms, when it's anaerobic respiration and no oxygen is present, fermentation is the next step. We'll go there first, and we'll follow that with aerobic respiration in the mitochondria. So fermentation happens in many types of bacteria that cannot live or do not live in the presence of oxygen, and also happens occasionally in our cells when there is not enough oxygen present. In this case, the NADH needs to release those electrons so that they can feed back into the cycle of glycolysis, and pyruvate, which was left over, needs to be recycled. It needs to end up going somewhere. And so there's lots of different ways in which this can happen, but a couple of the most common ones are what we'll take a look at now. Ethanol fermentation. This is where pyruvate is going to be converted into ethanol, and a molecule of CO2 is going to be released. Yeast would be one example of an organism that goes through ethanol fermentation. So when beer is being made, or when bread is being baked, fermentation is what's happening, and that CO2 that's being released is what creates the gas that causes the bread to rise, or the bubbles and the ethanol that's going to be in the beer. lactic acid fermentation, this is what's going to happen in animals. And in this case, pyruvate is going to be converted into lactate. That lactate will be converted further into lactic acid, and that acid will lower the pH of our blood. The process of fermentation, particularly by yeast, is probably the most commercially lucrative biological reaction. Everything from cheese to drinks to bread relies upon that process in order to produce the food that we know and love. Inputs and outputs, and moving forward. We proceed now to animals and plants and fungus and some protists that go through a process of aerobic respiration in the mitochondria. We start within the mitochondria. We have part of that cycle happening, we're beginning inside the inner membrane. This is known as the matrix of the mitochondria. And this is where we start with the citric acid cycle, which is also known as the Krebs cycle. What we start with, we had pyruvate. If you remember, pyruvate is going to be broken down and turned into acetyl-CoA. And acetyl-CoA is slightly smaller. So we went from a 3-carbon molecule to a 2-carbon molecule, so CO2 is a waste that's going to be released out from that process, and the acetyl-CoA is going to enter then into the next step, which is that citric acid cycle. So here's your inputs and outputs, but remember 2-pyruvate were created as a result of glycolysis, and so each of these steps is going to happen 2 times. So you see 2-pyruvate, 2 of the NAD+, making 2-acetyl-CoA, 2-CO2, 2-NADH. So those inputs and outputs are giving the total for each glucose. So the next step, that acetyl-CoA is going to be passed on and enter into the citric acid cycle. It's known as citric acid cycle because that's one of the intermediate molecules that the acetyl-CoA turns into once it gets into this particular series of reactions. So the main thing that you need to be concerned with here is what is being produced. The end goal is to make ATP. We make just a little bit of ATP here, but mostly what's happening is that molecules are being combined and molecules are being changed, and in that process electrons are being released, and those electrons are being picked up by NADH and FADH2 to be used later. Now again, because glucose was split into 2-pyruvate, for each glucose, this whole cycle is happening 2 times. Again, there's a complex step of reactions. If you study biology in college, you can learn all of these particular enzymes and names of molecules that would be produced. But for your purposes, just remember enzymes are important in this process because they're helping to catalyze and change this reaction from one thing to the next. Here is Mr. Krebs, who was the one who figured out the molecular formula and the steps of that entire cycle. Here's your inputs and outputs. Again, certainly know this information. Some of the things that are produced, more waste, CO2 is going to be created, and then we have the NADH and FADH2 are created along with an ATP. So a little bit of ATP is going to be produced in this part of the cycle. We go on from here to the final stage, oxidative phosphorylation. It's also known as the electron transport chain, also known as the electron transport system. So in this case, we start with electrons being released. Electrons along the membrane that is within the mitochondria are going to be passed from one protein to the next. As those electrons are passed, that some energy is being used to be able to pump protons from inside the membrane to outside of the membrane. Ultimately, oxygen is going to collect those electrons. It's going to combine them with some of those protons, which were pumped out, and make water. Water is going to be a waste product, but those protons are going to come in handy first before that happens. So here you can see there's a series of proteins that are embedded in the membrane, those are channels. You see that the protons, which are those hydrogen ions, positively charged, are being pumped out of the matrix of the mitochondria, and this arrows represent the movement of electrons. And so you can see electrons ultimately end up forming the water. Now, once you get all those protons on the outside, it creates a concentration gradient. So there's lots of protons outside, and they want to get back in. And so that's where the action really happens in terms of being able to make ATP, is when those hydrogens, those positively charged ions, start coming back into the matrix of the mitochondria. That process is known as chemiosmosis. And so this is when ATP are made. The protons diffuse back in, and when those protons diffuse in, this particular protein spins and causes it's an enzyme at the other end. That enzyme is going to help to combine the phosphates back with the ADP, and the bond is going to be reformed, creating ATP. So it's really the movement of hydrogen ions that transfers energy over to be able to form that ATP. Now there's an interesting connection here. On top, this shows what the ATP synthase molecule looks like, and on the bottom shows you a particular type of bacteria. And you can see how there is a similarity both in shape, and these colorations show different chains of different amino acid chains. And you see how that similarity is evidence showing that this particular protein at one point in a distant evolutionary past was a free-living bacteria that was co-opted by the cells, and they started to work together. So that's another connection to evolution. There's animation for you to take a look at. Again, look at the Prezi to be able to view that. Inputs and outputs, take a look. Okay, oxygen is necessary, so this is why it's an aerobic process. We see that these are all of the NADH and FADH2s that were created during both glycolysis and during the citric acid cycle. A lot of ATP produced along with some water. That water is a byproduct. It can be used by other cells in the future. All right, so just to sum things up, there is not necessarily a very specific amount of ATP that is produced, but what is important is to be able to see that there is a big increase in efficiency. When oxygen is used, it's going to be something like 19 times more efficient in terms of its ability to take that glucose molecule and produce some ATP from it. Now, in terms of harnessing energy, about 40% of the energy in that glucose is successfully harnessed and converted into ATP. But a lot of other fuels, if you look at, for example, gasoline in a car, the percentage of energy use is much lower. So this is, even though a lot of energy is lost, it's pretty normal for a lot of energy to be lost in a lot of these processes. So that's a pretty remarkable feat of biology for cells to be able to actually get out so much of that energy from the sugar that we take in. And one final connection into some of the things that we'll be studying in this unit, there are actually multiple ways in which different molecules can enter into the cycle. So carbohydrates, like glucose, can go through the cycle from beginning to end, like we mentioned, like I went through in this video. But fats can be broken down into glycerol, which can enter into an intermediate step of glycolysis, and fatty acids can be broken down to make acetyl-CoA and enter in at a much later point in the cycle. Proteins will be broken into individual amino acids, which can further be broken down to make pyruvate, to make acetyl-CoA, or to make different molecules within the citric acid cycle, and that allows us to be able to use all three of those, proteins, carbohydrates, and fats, as potential sources of energy. So in digestion, we can see that all of the different molecules that are in our food ultimately can enter into some point in this process and ultimately make ATP for us.